Biological diversity, or biodiversity, is the variety of all life on Earth. It supports healthy ecosystems and food webs that are resilient to the ongoing effects of climate change and contributes to human health and well-being. To many, biodiversity may only be a list of species, but it is far more than that. It encompasses 3 levels: genetic, species, and ecosystem diversity. Genetic diversity is the variety of genes within a species. Species diversity involves the number and variety of species within a habitat. And ecosystem diversity considers the array of habitats within a landscape. Biodiversity is not merely the sum of these components, but also includes processes and complex interactions among them. Genetic diversity within a species is crucial to its resilience, or its ability to survive and adapt to environmental and biological challenges. Without resilient species, ecosystems become destabilized, leading to catastrophic consequences for nature and people. This interconnected relationship underscores the critical importance of genetics as a foundational component of biodiversity that needs to be conserved. However, it is not only the presence of genetic diversity that is important, but also the functional processes that create it and shape its patterns within a species, like mutation and the amount of natural gene flow between populations. Therefore, genetic diversity within and between individuals and populations and the processes underlying it must also be considered in conservation. Genetics, which is at the core of biodiversity, has often been overlooked in conservation efforts and legislation the world over. The latest international agreement on biodiversity, the Global Biodiversity Framework, finally now emphasizes the importance of the inclusion of genetic diversity in conservation monitoring and reporting. Through the process of natural selection that acts on the genetic diversity within a species, populations in different areas adapt to their environment, a process referred to as local adaptation. In addition, populations that are geographically isolated, for example by a river or mountain range that prevents gene flow, accumulates genetic differences over thousands to millions of years, and this leads to distinct populations that may potentially become different species. Looking at the mouse example, the colors represent genetic diversity. Some populations have more genetic diversity, and some contain genetic variation not present in any others. Therefore, when we detect that populations are genetically distinct due to evolutionary processes, it is important to conserve them separately so that evolution may continue its natural course. Conservation geneticists call these genetically distinct populations evolutionarily significant units, or ESUs for short, because they are populations or units of a species that are evolving independently from each other. This is an important term that we will use throughout the rest of the presentation. For some species, ESUs might correspond well with the described species or subspecies, for example in giraffe and sable antelope, while for others like hartebeest, traditional subspecies classifications have not kept pace with new insights provided by genetic data, which is continually becoming more affordable to generate. Principles of maintaining evolutionary processes. Translocations can easily disrupt these evolutionary processes and break down the genetic patterns. It is therefore critical that all authorities and stakeholders involved in translocations have the same understanding of evolutionary processes, genetic structure, and the consequences of ignoring these in translocation decisions. Therefore, the African section of the IUCN Conservation Genetics Specialist Group, Congen Africa, together with local and regional conservation authorities and researchers, have developed species-specific genetic management guidelines for commonly translocated species that have sufficient genetic data available. However, many species do not have sufficient genetic data to develop such guidelines, and therefore we have also developed general genetic principles to guide translocations in such cases. There are 4 main genetic principles to consider when making translocation permit decisions that will ensure sustainable management of species for conservation and the biodiversity economy. Adopting these principles will help maintain evolutionary processes and the genetic patterns that have evolved over thousands to millions of years, which is crucial for species' long-term survival. Wildlife is often present in fragmented populations due to human interference, and we therefore need more hands-on approaches to manage species as exemplified by these principles. Principle 1. Translocations should mimic or reinstate natural processes that have led to the current day observed genetic diversity and structure within species. This means that translocations should mimic the natural movement or dispersal of individuals, allowing for gene flow between populations. Where gene flow has been disrupted, for example, a city now dissects 2 populations of a species, or populations are now fenced, then translocations should serve to reinstate this process. Principle 2. Translocations must not break down existing genetic structure or ongoing evolutionary processes. This means that translocations between different ESUs must not occur, and existing mixed ESU populations should be managed separately from populations containing a single ESU. The exception to this is when a population has been deemed to be threatened by inbreeding and requires genetic rescue. Only if there is no other suitable population within the same ESU from which to source individuals for genetic rescue may another ESU be considered as the source. Genetic diversity must also be measured against the natural genetic diversity of a population, and more is better is not true if it is a result of inappropriate mixing. Principle 3. If translocations are planned outside a species' natural range, then individuals should be sourced from a site in the natural range of the species closest to the introduction site. This is contingent on the habitat where the species is to be introduced having been deemed suitable for the species, so that individuals do not suffer due to maladaptation to their new environment. Principle 4. Captive populations that have been selectively bred for certain traits should not be mixed with wild populations. For example, animals bred for different coat colors or longer horns. In some instances, selectively bred individuals may have high levels of inbreeding and could pass on as yet undetected harmful traits to offspring. Even though to the human eye they may appear to have superior characteristics. Species-specific examples of genetic management guidelines. The remainder of this presentation will illustrate how the 4 principles should be applied to different species that are commonly translocated in southern Africa. To illustrate these principles and the consequences of permit decisions for the translocation of animals, we will use two examples with species-specific guidelines: sable antelope and leopard. The sable antelope has 5 ESUs. They are the West Tanzanian, Eastern, Angolan, Zambian, and Southern ESUs. The distribution of each ESU is shown by a different color, and together they show the distribution of the species as a whole. These ESUs formed as a result of geographical barriers that have existed for thousands of years, such as the Zambezi River. The Zambian ESU is only present north of the Zambezi, and the southern ESU only occurs south of the river. In the evolutionary history of sable antelope, the Zambezi River has acted and continues to act as a barrier to gene flow, so that genetically distinct populations have developed on opposite sides of the river. These populations have diverged sufficiently over long periods of time, potentially hundreds of thousands of years, to be considered separate ESUs. According to the principles mentioned before, translocations between ESUs must not occur because they will risk destroying the natural genetic structure that is formed as a result of this river barrier. If animals are translocated from one ESU into the range of another ESU and individuals subsequently escape or are deliberately released into the wild, they can breed with animals of the native ESU. This breakdown of the natural genetic patterns could lead to negative consequences, such as reduced local adaptation or fitness of the indigenous sable antelope population, or an increased chance of local extinctions leading to destabilization of the ecosystem. Even if risk to the species cannot be demonstrated, the loss of genetic structure and breakdown of genetic processes represents a loss of biodiversity. If mixing of ESUs has already occurred, a genetic test for all individuals prior to translocation will be required. Permits should only be issued for the import of sable antelope from countries or localities containing the southern ESU, and not from any areas where other ESUs exist. Similarly, permits should not be issued to export southern ESU animals to countries or areas where other ESUs exist, as this will result in loss of biodiversity in those areas. Within each ESU, we might have distinct genetic subgroups. These subgroups are called genetic management units, which are not as different from each other as ESUs, but they still represent distinct genetic units within an ESU. Over evolutionary time, there is some gene flow between these units, which means they are genetically more similar. Consequently, the biodiversity risk of mixing animals from different units is usually lower than it is for ESUs. Nonetheless, in the case of sable antelope, the guidelines are to avoid mixing genetic management units through translocations. When translocations must occur, then animals must be sourced from a population in the same ESU and as far as possible from the same genetic management unit as the receiving population or area. While different ESUs are shown in different colors, different genetic management units are indicated on the map by the different symbols of the same color. The location of each symbol indicates where actual genetic data was collected and used to determine which ESU and genetic management units the population belongs to. There are 3 genetic management units within the southern ESU, namely the East Southern, shown as green circles, mid-southern, shown as green squares, and west-southern, shown as green pentagons. If a new population is to be established in South Africa within their natural distribution, then the animals should be sourced from the mid-southern unit, as this is the naturally occurring unit within South Africa. Acknowledging that sable antelope from non-indigenous ESUs and genetic management units have already been brought into South Africa, it is therefore very important that sable antelope be genetically tested to determine which ESU and genetic management units they are part of before being moved. Animals from non-indigenous ESUs or genetic management units or animals of mixed origin should always be kept separate from indigenous populations. In the second example, we will look at leopard. There is only one recognized subspecies in Africa. There is high genetic diversity with few physical barriers to their movement. Genetic structure across the continent generally follows an isolation by distance pattern, where animals further apart are less similar than those closer together, but there are no distinct groupings. However, there is a mainly southern African mitochondrial grouping, indicated by green circles, with a more African-wide grouping indicated by gold circles. The contact zone between these groupings is in the eastern part of South Africa. Until we fully understand the natural processes that shaped the genetic structure, a precautionary approach should be adopted. Management of leopard populations in southern Africa should thus allow for and support the natural dispersal of individuals, and translocations over longer distances should be avoided. It is suggested that individual leopards should not be moved more than a maximum distance of 200 kilometers and not across the contact zone. Now that you have seen how the genetic principles can and should be applied in the practical examples of sable antelope and leopard, it is important to recap a few key take-home messages that will assist when making translocation and permit decisions. In southern Africa, the wildlife economy, based on sustainable utilization, is a crucial economic driver and job creator. However, for the use of wildlife to be considered truly sustainable and responsible, and for species conservation programs to be more effective, only animals from appropriate genetic units or locations should be mixed. Incorporating genetic considerations into translocation decisions is therefore critical. It is hoped that the guidelines developed will assist in translocation decision-making by synthesizing otherwise obscure and difficult to understand genetic information from multiple sources into simple and easy to understand maps. The species-specific guidelines and the general genetic principles for translocations are available on www.congenafrica.com. As new data becomes available, the guidelines will be updated. Because our understanding of genetics is rapidly advancing, it is recommended that the latest guidelines are always consulted prior to making translocation decisions.